This demonic nanorobot technology is not only placed in vaccines, where they found to be around 1 million nanobots in one droplet of the vaccine, but it is also placed now in anesthetics, in particular in dental anesthetics, and they even put it in insulin, as was proven scientifically. Look at that.
Today, I want to present to you an investigation of Lantus long-acting insulin that was sent to me. I am going to share my screen. So in this investigation, I want to present that the makers of the Lantus SolarStar pen, which is Sanofi, had created an assembly plant in China. And this is important because there are certainly concerns about manufacturing processes in China and what effect that might have on American civilians. Sanofi also helped make 100 million Pfizer BioNTech vaccine doses for the COVID-19 bioweapon. In this slide, I want to explain that it's long been known that advanced hydrogels are being used for the controlled delivery of insulin. So hydrogel is a programmable matter that can self-assembly from a nanoscale to centimeter size in minutes. In my research, I have found that it's a polymer hydrogel that is actually forming in the C19 injected as well as in unvaccinated individuals via shedding and environmental contamination. And then these hydrogels are creating rubbery clots. For further information on this research, please refer to my Substack, Anna Mihalcha, MD, PhD.substack.com. In this article, it is said that the use of 3D hydrogels has been used with much interest for various biomedical applications. Hydrogels can mimic the extracellular matrix. So that means the extracellular space in our body can be mimicked by this hydrogel. And so it can retain large quantities of water with tunable properties. Tunable also could be exchanged into programmable properties. So all of life acts through resonant frequencies, and these frequencies via biophotonic light, via phonons, which are sound waves, are what are in control of the communications between cellular structures. So it says further, which renders them suitable for administering a wide range of sensitive therapeutics. Several studies have demonstrated the fixation of insulin within the structural mesh of hydrogel as a bioscaffold for the controlled delivery of insulin. The reason why this wording is very important is because the structural mesh is exactly what has been showing up in people's live blood. And in our analysis via near-infrared spectroscopy, we clearly showed that these are polymers like polyethylene glycol and other derivatives like alkenes and polyenes, as well as polyamides. So this mesh is clearly visible in the live blood, and I've shown this in my research, and it's important to understand this background for what I have seen in the Lantus insulin. So this is the information from the Sanofi website on what is in the Lantus injections. And if you can see here, a 10 milliliter vial presentation contains the following inactive ingredients. There's M-cresol, 2.7 milligrams, 20 milligrams of glycerol, and 12 micrograms of polysorbate 20. And I just want to review what these chemical compounds are and what they do. Additionally, hydrochloric acid and sodium hydroxide are used, and the pH is very acidic at only about 4. Glycerol, which is one of the components in the Lantus injection, has been shown to be part of hydrogels for sensing applications. So hydrogel can be used to biomimic tissues, but also to create soft robotics. Soft sensing technologies have the potential to revolutionize wearable devices, haptic interfaces, and robotic systems, it says. The work reports the development of a versatile ionic gelatin glycerol hydrogel for soft sensing applications. Soft sensing means something is measuring your physiological processes and sending that information to the cloud, to a receiver for biomonitoring. The resulting sensing device is inexpensive and easy to manufacture, is self-healable at room temperature, can undergo strains of up to 454%, presents stability over long periods of time, and is biocompatible and supposedly biodegradable. I want to comment on this because we've done extensive studies with the clots of deceased as well as those that are forming in the COVID-19 injected as well as unvaccinated people. And that's a rubbery material that we could not dissolve with 9 solvents, including things like toluene, lye, and different acids. So this is very important to understand that we have tried to degrade these things, and the most corrosive chemicals cannot do it. Polysorbate 20, which is another ingredient of the Lantus injections, is a 20 repeat units of polyethylene glycol. Polyethylene glycol has been used as the hydrogel lipid nanoparticle platform for the Pfizer COVID-19 injections and bioweapons. M-crazole is a chemical compound that's highly toxic. So there is a hazard to health which can cause serious or permanent injury. It's flammable, and it's considered a DOT hazard level as a poison and a corrosive. The hazards are via inhalation, it can cause mucosal irritation and systemic poisoning. For the eyes, there can be intense irritation and pain, swelling of the conjunctiva, corneal damage. In the skin, there can be intense burning, loss of feeling, wrinkling, white discoloration, and softening, and gangrene may occur. When it's ingested, there can be a burning sensation in the mouth and esophagus. Vomiting may result. Acute exposure by all roads may cause muscular weakness, gastroenteric disturbances, severe depression, and collapse. Effects are primarily on the central nervous system and edema of the lungs. Injury of the spleen and the pancreas may occur. So here are alternative chemical names, which are, for example, 1-hydroxy-3-methylbenzene. Benzene is known to be toxic to human, hydroxytoluene. And then I picked up just a definition from good old Wikipedia. And what it's saying is that metacresol is an organic compound that is made traditionally extracted from coal tar, and these are obtained by the production of coke from coal. Pegylated hydrogels are used for the controlled release of biomolecules in regenerative medicine. So polyethylene glycol specifically, which has huge concerns for anaphylaxis and allergic reaction, is used in a wide variety of biomedical application, as well as to create these scaffolds for regenerative medicine. So you can load biomolecules with these pegylated hydrogels. And so this is important because you will see how the molecules within the Lantus will self-assemble. For more evidence and more background information, please refer to this video because it shows extensive research on what is happening with the blood, where we have in live blood seen how these filamental structures self-assemble, and we have proven that these are made from hydrogel that are literally programmed into bacteria, archaea, and eukaryotes, all 3 domains of life, which are programmed to make these polymers and have them self-assemble. This is an important background video. I was part of an international team that looked at the contents of the Pfizer vial COVID-19 bioweapon. And the first time that I looked at it via Zoom with my colleague, Dr. David Nixon in Australia, this is what we saw. What I want to bring your attention to are these blinking lights that are changing colors. Those are quantum dot-like structures, and they have been found in the blood, and you will find them in the vials. You can see an enormous amount of movement here of metallic-looking objects that are assembling this metal-like appearing structure. And you can see at a higher magnification again, here are these spherical structures that are blinking. You can see how there's self-assembly going on. You can see many of these other longitudinal type of metal appearing things. So this is important to understand, because when I show you the Lantus, we want to draw the parallel between the COVID-19 bioweapon that has many undisclosed ingredients, including heavy metals, and then the pegylated lipid nanoparticles that create self-assembling hydrogel and biosensors and microchips that have been found by Dr. David Nixon. So again, you can see that these blinking lights, they have blue lights, there's red lights, golden lights that just show up, and there's a tremendous amount of movement and coordinated self-assembly of this structure. So as we are observing it, you can again see these blue blinking dots, and they become part of the structure, and they also create these filaments that we see in the blood. So it's very important that people are aware that there is this parallel between the COVID-19 injection and what I'm about to show you in Lantus insulin. So this is when I put the Lantus insulin on a slide, and over time I was just observing and photographing what was happening. I have a dark field microscope, and you can see how from the solution, once it's drying out, that there are multiple structures visible. This is hydrogel that's beginning to assemble and structure, and then you can see the background here with many of these blinking lights that are literally parallels to quantum dot technology. I believe that's what they are, because they change colors from blue to red to green. They're used for biosensing applications. They also are used in quantum applications. They work as a battery. They have a toxic heavy metal within them. And then you can see here what appears to be a spherical bubble with multiple smaller spheres in them. I have actually proven extensively that this is part of the self-assembling mechanism of hydrogels. here you can see how the background has solidified with many very small particles. Looks very similar to what we saw in the COVID-19 bioweapon. And here you can see how a filament has formed. These are the same filaments that we see in live blood and that we've identified to form the rubbery clots that we've identified in the deceased, in vaccinated as well as unvaccinated from shedding and environmental resources. Right now, humanity is under a full-bore assault. We found these filaments sprayed via geoengineering, as well as the metals are being sprayed. We found them in other medications, for example, dexamethasone. It's well known that hydrogels are being used for biomedical applications. Here you can see that the hydrogel is assembling, and here you can see that a structure has formed out of the background. These are further images over time, as you can see that dense structures are being created, as well as a background of material that is classic hydrogel. Here you can see how these filaments that we see in the blood are being formed. You can see the spheres that are part of the assembly process. This is a higher magnification of the same ribbon. Here you can see that a sphere has solidified, and you can see many of these little dots that are exactly these blinking lights that we were seeing in the COVID-19 bioweapon that are now have coagulated together to start forming structures. Here you can see in the background that self-assembly is taking place. The bluish dots are these quantum dot technology, and then you can see that the hydrogel is forming and coagulating in multiple areas. Here you can see more complex structures. Again, these spheres are the quantum dot technology. They are creating and assembling here with a complex structure and a mesh-like fiber. Remember the scaffolding that was mentioned in the literature about the hydrogels that would mimic the extracellular matrix. So literally, this is what is happening here, a dense meshwork that is intercepted by these quantum dots that are artificially intelligent and are able to direct the self-assembly. And you can see that more and more material of hydrogel is being produced and coagulated. Here you can see the background. Again, many spheres are visible, and you can see how the hydrogel is forming on the back of the on the slide and becoming denser. Here you can see how many areas of the hydrogel production have happened. Here is a filament that's being built or self-assembled, as well as many other of these filaments that we've also seen in the blood. People always ask me regarding graphene oxide because there are certain groups like La Quinta Columna that calls the finished product of these filaments graphene oxide. We've chemically analyzed the blood as well as the clots, and we found that these are polymer hydrogel, very close akin to rubber. So these are polyamides and alkenes and polyenes, which is polyethylene glycol. So that is the actual self-assembling hydrogel that can grow. Within it can be graphene oxide or carbon nanotube, as well as heavy metals that then determine what type of electronic device or nanorobot is being created. The hydrogels can also be used for the quantum dot self-assembling technology. Here you can see that a very dense area here already has built. You can see many of those blinking lights. A lot of material has self-assembled, and I am just showing on a 10 times objective just how much of this material has been shown and is self-assembling here. So all of these structures are in the process of creating filaments, of creating denser material, and they're doing so from a nanoscale that I'm not able to see from a microscope. But you can also see these bluish spheres that are changing colors from blue to red. So these are quantum dot-like structures, and then more and more material that is over time accumulating. The total time that I spent looking at this particular liquid during self-assembling was 3.5 hours. So here what we can see is a higher magnification. You could see that more and more material is being self-assembled and creating more structures. And you can see here how there are mobile elements of these lights. And remember, this is a drug that should be sterile, so this cannot be anything like a bacteria, particularly because these bacteria aren't nanorobotics or artificial intelligence. Here I am endeavoring to show you while rotating the microscope objective. You can see that there's a bluish sphere that's traveling rather quickly here, and that you can see that all of these materials are created by the spheres that have coagulated and replicated themselves and self-assembled. So imagine that this self-assembly process is happening in the human body. This is how then microclotting starts to occur, and a much more denser filament network is being created. And you can clearly see that these things are mobile. They're moving, and they're spherical in nature, and they can be emitting light signatures that are very specific. So you can again see here significant movement of individual aspects of these. These are literally nanorobots that are self-assembling, and they have an intelligent design because their objective is to create this hydrogel scaffold matrix. And so here I am showing you more of this sphere. As you can see, it has, like a Russian doll, many different spheres within the spheres, and here's a larger sphere. And what is assembling within that is literally creating these filaments as well as the substrate. And you can see that a lot of hydrogel has been self-assembled and is now becoming denser and denser. You can see in between those spherical things that are again like these blinking lights. And you can see many of these round spheres that I have documented in my research to be actually the source of creating the hydrogel filaments. And you can see here another, this is literally a construction site, and I have shown this in detail. So what I can see here is that many filaments will be assembled, and you can see that the background hydrogel is becoming quite dense. Many construction sites are available, and right here you can see that the substrate is becoming denser and denser on the slide, and continuing self-assembly as it is drying out. But you will see that there's huge activity going on, and that there's very, very intense self-assembly going on. So if I show you a little bit here, you can see these bluish lights, and this is continued self-assembly, continued denser material. And again, because this substrate would be injected on a day-to-day basis in a human, these things can create microclotting, they can create problems within the body. It says that this stuff is biodegradable, but you can see that its tendency is to continue to self-assemble. So here what you can see is a video of some of the images that I showed you earlier. And now it's visible that on the slide, more and more denser areas. You can see here that filament growth is proceeding. And you can see a lot of movement going on. This is little tiny particles that are working on the self-assembly and that are going to different places to then become self-growing clots. And this is exactly the same filaments that we see in human blood. And that has been transforming the blood, that has caused extreme rouleau formation, it has caused blood clotting, and we also, in my work with Clifford Carnicom, have clearly shown that the electrical conductivity of the human blood has been significantly decreased, which means that the life force literally has been hijacked. Electricity is how all of life functions. So you can see this very active hydrogel self-assembly going on, and how these particles that are clearly blinking lights that are changing colors from blue to red are working and traveling in an intelligent manner to assemble these things, and they're growing themselves. They're emitting light, which is the optical communication between them to coordinate this very intricate and construction site. You can see this is absolutely intelligent, and as I'm showing you, I'm keeping it just a little bit out of focus here on the background so you can see the blinking lights better, and literally the spherical content of quantum dot technology that is working around this filament, as you can see here, to build it and to deposit more hydrogel. If I speed this up, you can see the tremendous amount of motion that is visible here. But even like this, so imagine this is a pharmaceutical medication, and it is assembling something that could cause a clot, that could cause a heart attack, strokes, as well as all kinds of other accelerated aging, microvascular disease, and that is in every insulin syringe. I've looked at other insulins, it's also in there. So the long-acting insulins are all using hydrogels to make them time-release bioavailable. You can see again here the many bluish components, but also that the quantum dots change from blue to red to yellow. And over time, this filament continues to grow, and the background also continues to grow. So this is a video of what I showed you earlier of the slide. And you can see how the filament network, the scaffolding that was discussed earlier. So imagine this could be your extracellular matrix that is literally being mimicked. So when this stuff is injected, it is fusing with the human body, with human cells. It's mimicking our natural organ system, and it's using these quantum dots, which are biosensors and biorobots, to create this matrix that then can be externally via frequency controlled. So this is all part of the literature of nanotechnology and synthetic biology. But you can see that I filmed this assembly, and imagine that something like that would assemble in the human body and, for example, replace the brain or replace any other organ microscopically, that it could clearly interfere with the function, cause substantial problems. And this is not something that humanity wants in their body. And I don't think that humans are understanding that this drug delivery system has been used, and it is literally the platform to create cyborgs. You can see that if I play with the focus, how these devices with the spheres, the quantum dot technology is visible in red and blue, and how they've been used within this matrix to create this the scaffolding of the hydrogels. So here you can see, the much denser self-assembly of the hydrogel and many components. And but I wanted to show you that certainly the scaffolding mesh is within the drug itself. Here I'm showing you different phases of the construction. So you can see this round area where the hydrogel is precipitating, is being self-assembled. And then here the center with all of these filaments, and then with this quantum dot technology. In this video, I am showing also how these filaments that we usually see in the blood are also being created, so just from the drying out process. But as you can see, even though this is now dried out, clearly there's still a construction process, there's still movement going on, and I'll show you in different areas much more intensive self-assembly. But in terms of, these are exactly the types of things that we see in the blood. And so now that you see video footage, you are understanding that the self-assembly is happening in multitudinous ways. This is just one drop of this Lantus insulin. You can see an image from the sphere that I mentioned earlier. This is classic of what we see now in the blood in almost everyone. You can see the bluish quantum dots in there, and then the self-assembly of the hydrogel. And here's the ribbon being constructed, and these are the filaments that we've seen. When we worked with the Pfizer vials, Dr. David Nixon showed that these are optical communication cables between microchips that develop. Here's another image in regards to that, and a video just showing you so that you can see how these bluish quantum dots are helping to self-assemble from this sphere, and then it is growing this filamental structure. So this doesn't stop, it just continues and continues. And some of the filaments that I've seen in the blood come out of one of these spheres is thousands of times larger than a red blood cell, so it can definitely produce clotting. Here you can see a very active construction site. You can see over here how hydrogel has been solidified. And this is how all of these little nano robots that have self-assembled to larger size so that we can see them with the microscope are building this this hydrogel structure, which is the same thing that makes up the clots. We have proven that chemically. We are near infrared spectroscopy. We've analyzed the clots. It is absolutely the same. We've analyzed its microscopy. We isolated the origin of genesis, which are these cross-domain bacteria. So this is synthetic biology that is creating hydrogel. and it's also moving. And you can see how dense this network is that has been created and is still being created. Here the construction has progressed. You can see that many of those bluish dots are part of this. And this is a very dense filament here that's been created in a large structure from the hydrogel that has self-assembled. So clearly, if this is in somebody's blood, right now the magnification is about 2,000 times. You can see how from the very tiny and the very small, all of these large structures are being assembled, and just the amount of intelligent assembly and movement and coordination of the light communication here, the different colors that are being emitted, and that is an intelligent way that these nanorobots and quantum dots are able to communicate with each other and signal in a coordinated way how to build these larger structures. And in a moment, I'm going to show you exactly what that looks like. But here, again, in one drop of Lantus, which some people inject twice a day for decades and decades, you can see how much artificial intelligence, synthetic biology, and nanotechnology they're injecting. And so this can cause substantial health effects, and it's transforming these diabetics into something that is part biology and part technology. And it's literally replacing their organs as well as their systems. And the reason why I'm showing you all this video footage is just the enormous extent of activity and self-assembly that's growing from the nanoscale. So initially this wasn't visible, but now it's been about an hour and a half that I've observed this, and you can see how active this self-assembly is everywhere on the slide, and how it is growing these larger structures that we've seen earlier and we will see again. I just wanted to show you how these quantum dot blinking lights are creating and coordinating self-assembly here. And again, the ramifications of this is enormous, because how many diabetics are there who are insulin dependent? We've seen this in dexamethasone. I've looked at Benadryl. I found it. My colleagues have found self-assembling nanotechnology in anesthetics. So these commercial drugs are literally weaponized against human. You can see this enormous light activity here, clearly signaling very, very intensely. And so this is their communications. These little robots do not talk. They give off light signals at specific frequencies, and this is how they coordinate the self-assembly. And they're spherical in nature and are able to become very complex and then assemble themselves to become large hydrogel structures. And again, all of this I see as a finished product in the blood, and it continues to grow. I've also looked at embalmed blood of an individual who's been dead for 8 months, and the process of self-assembly continues even though the person is deceased. So here I want to show you again one of those areas that now has created these filamental-like structures. And what this is, it's a video of showing you a close-up of how these filaments are beginning to be assembled, and then how busy the construction site is. So here you can see that many of these little islands, they continue to be assembled. You can see that these are those bluish blinking lights actually become the substrate of the filaments. And you can see how extremely active these light signals are. Here you can see a denser material. Some of them are moving rapidly, some aren't, but they are literally becoming the filaments and growing the clots. So it's these bluish aspects that are creating this. And here you can see the self-assembly. So aspects of hydrogel have been created, and you can see it's being created here and here, and literally like an ant colony, they are transporting pieces of this hydrogel to then assemble and grow. So you see right here, a piece has been attached. There are all of these nanorobots that are working to attach it to this larger piece of hydrogel. It's going on everywhere. You can see the transport here, literally moving like an ant colony, but absolutely intelligently coordinated. This is artificial intelligence that's growing from a quantum level, and that's communicating via optical communication. And you can see now literally documented self-assembly. You can see here a large hydrogel clot being assembled. That's exactly what it's looking like on microscopy, and I have so much evidence of how it looks like. But see here how smaller and smaller pieces are being transported. Again, I'm showing the blinking lights are doing the job, and they are working together in a coordinated fashion to grow this hydrogel, and they're transporting pieces of it. They continue to self-assemble them, and then in an intelligent way are continuing to grow what are these polymer clots. These same blinking lights, I have absolute evidence in human blood, how they use the substrate of the blood to literally make the building blocks to create the mesh network, and to replace the organ of blood with hydrogel rubber. And I've also done testing where I just left the blood of even unvaccinated people who weren't on insulin, haven't taken the shot, just from environmental shedding, who clearly had this rubbery clot that's cannot be disintegrated mechanically, and no blood thinner will dissolve it. EDTA and vitamin C help the assembly, so prohibit the assembly process. But if you can see how much of these blinking lights and nanorobots there are, you need a huge amount of EDTA and a huge amount of vitamin C, particularly. So the more you're injecting this insulin, the more you're giving the COVID-19 bioweapons, the more stuff of these electronic artificial intelligent devices you have in your body, and they self-assemble like a weapon system and nanorobotics swarm, and they just grow and take over the body. And you can see that in one drop of Lantus, all of this activity is going on. This looks exactly like the Pfizer vial that I had looked at with Dr. David Nixon, and you can see how literally the self-assembly process is going on here. You know, pieces of hydrogel are being attached, they're being glued together. You know, other pieces are being transported, they're being built. This is how the clot is being generated. And this is very powerful evidence that literally Lantus insulin is a weapon, not a medication, that is creating self-assembled clots that might kill people, as well as cause substantial illness, heart attack, stroke, and other problems. The level of activity of self-assembly here is just absolutely astounding. It's horrifying beyond belief. And this is about 2 hours in of me just observing this self-assembly process. It's not drying out. You can see that despite the fact that whatever water was there is evaporated, the construction continues. And that's also what we've noticed with Dr. David Nixon's work, is that the self-assembly process, the hydrogel itself, just continues to grow. Literally, I mean, the amount of activity seen here is enormous. And these structures grow larger and larger until they're, you know, Richard Hirschman sent me a clot that was 33 inches long. It doesn't stop, and it doesn't stop at the death of the host. So I believe that this is one of the other sources in which people get contaminated, in which things get a lot worse. These medications that are created by the same perpetrators are weapons. And you can see here very clearly how the hydrogel has been assembled, how this process is going on. These huge pieces are being attached here to grow the clot, to grow this hydrogel island that then would, if it's floating around in your vascular system, would cause plugging up of the arteries, strokes, and death. You can see within it, the constructions are still going on in thickness and dimension. And you can see the bluish hue of the robots. Here you can also see how it is working to assemble this. And I mean, this is enormous. These are crimes against humanity that are unfathomable.